invited Prof. Charles Pineau from French National Institute of the Health and Medical Research as the speaker today. Also joining us in the panel discussion will be the Dr. Chen Chang from the National Center for Protein Science Beijing, Dr. Tiannan Guo from Westlake University, and I'm Ray Sun, a postdoc from Omics Lab at Westlake University. I'm very pleased to have this opportunity to moderate this webinar and to join the panel discussion. I hope you will enjoy this event. And Prof. Charles Pineau has been working in the field of reproductive biology, and it's a renowned specialist of spermatogenesis. He has heavily invested in the development of proteomics and integrative proteomics technology, and he's applying this to answer biological and clinical relevant problems in the field of testicular pathologist and reproductive toxicology. Dr. Pineau currently served as research director and team leader at INSERM Unit 10185. And he is leading the proteome faculty, and he is the director of BioSeed. Dr. Pineau's team focuses on the deciphering of the testicular proteome in mammals, with a priority given to the identification of novel germ cell proteins, and play a key role in the normal and pathologic spermatogenesis. His recent breakthrough work identifies novel testicular germ cell proteins using a proteogenomics approach. His team is part of the Chromosome Central Human Proteome project and is tracking the characterism of missing protein in the human spermatology. Currently, he is VP of the Society for Reproductive Medicine, PI of the French International Initiative in the CHPP, and he is the new charm of the Human Proteome project. His talk title today is the Human Proteome HPP Ground Challenge Project. Now, before we move to the talks, please also allow me to introduce all the panelists. Dr. Chen Chang is an associate researcher of the National Center for Protein Sciences, Beijing. He is engaged in proteomics and bioinformatics research and has developed the proteomic data quantification software PANDA, the label-free quantification algorithm LFAQ, the multi-omic analysis platform ExpressWes, along with other large-scale data analysis algorithm and software, all with an independent popularized. His work has been published in journals such as Nucleic Acid Research, Bioinformatics, AC. He was awarded the first Prime of Invention and Innovation Award of the China Association of Invention in 2020. And next is Dr. Tiannan Guo. He is tenured associate professor at Westlake University, where he served the director of the iMarker Lab. He is also the founder of Westlake Omics. Dr. Guo leads the Omics team, which is dedicated into developing and applying proteomics methodologies to generate large-scale proteomic datasets, with the ultimate goal of facilitating our understanding of biology and disease. The team is building an intelligent engine that will analyze this data and provide valuable insights. And this is me. I received my undergraduate and master degree in pharmacology from China Pharmaceutical University, and my PhD degree in biology from Westlake University, Zhejiang Westlake University Joint Training Program. During my PhD, my research interests are precise molecular classification and the prognosis determination of various diseases based on proteomics. In July 2022, I joined the Gomics lab at Westlake University as a postdoc, focusing on interference-based functional proteomics research.
Okay, so how long do I have for my presentation? I may have to skip a few slides. Just tell me.
Yeah. So normally it's about 30 to 40 minutes. But okay. Maybe if there's any questions, I may ask in between, or some other people may ask in between. After that, we have no discussion.
Okay, so I'll start a chronometer, and if I go too long, I will skip a few slides. So okay. Thanks again for your invitation and for giving me the opportunity to present the HPP Grand Challenge. And I will start with a couple of information that I was telling you, young colleagues, earlier, 10 minutes ago, that this may be some basics, but people have to keep in mind that, well, there's already decades that the first genome has been sequenced. It started with E. coli, and then the yeast, and then the Drosophila. And you can see from the above images that the number of genes increases with the complexity of the organism. You know, an E. coli doesn't resemble a red fly, you know. But basically, if you go to the bottom, you have C. elegans, 19-something gene, the mouse, 18-something gene, and the human, roughly 20,000 genes. And basically, with an average number of genes, which is really the same, the individual don't look alike. You know, it's really different. And what makes the difference is the protein, not the gene. And I took this slide from my old colleague, Amos Berok, from the SIB in Geneva. He presented this years ago, and is still valuable for roughly in the human 20,000 protein coding genes. It does not correspond to 20,000 transcript, nor that it corresponds to 20,000 proteins. So you have alternate splicing of mRNA, you can multiply by 2 to 5, so which makes something like 50 to 100,000 mRNAs. And then you don't stop there, because you have what we call the post-translational modification of protein. We know roughly 70 different PTMs at the moment. So if you multiply 50 to 100,000 transcript, it gives something like 5 million proteins. And we still don't know at the moment whether we are optimistic or pessimistic. It's probably more than 5 million protein coming up from 20,000 genes. And why? It's just, for example, for those of us, and I'm not including myself here, who were in the university and back in the 1950s, you all remember, at least by reading this great work at the moment by Biddle and Tatum, and they would say one gene corresponds to one protein. That does not exist anymore. Roughly in the eukaryotes, one gene encodes for about 7 different proteins, which now you can start to understand why it's beginning complex. But this is not enough. And you look at NextProj, which is the international repository for human proteins, and you can see that you have something like 42,000 isoform. We have something like more than almost 1 million identifiers, interaction, links between protein and term, these big numbers here. And recently, and we see this in Cancun, end of last year, those groups are coming up with small and small peptides that are encoded by these small open reading frames. We have something like over 6,000 of these teeny proteins that are real life and that were identified. So it's beginning complex. And then we have what we call the proteoforms. For the same protein, just a teeny modification on the surface of PTM, phosphorylation, acetylation. This is like a donut, you know. This is the image I could find. You have a tremendous number of proteins that could be modified just by adding a few features on it. So it's beginning really complex, and we don't take into consideration what we call the microbiome. In the human body, 1 to 3% of the body mass is about 46 million genes encode for proteins. And some of these can be useful for the physiology of the host. So imagine basically the bacteria in your gut. We know that some of these bacteria produce some protein, and among this protein, some are known to be used by the human physiology for the organism to work. There's plenty of examples in the nose, in the mouth, in the colon, in the stomach, in the skin, in sexual organs. So these proteins are not encoded by your own genome. They're encoded by some organisms that is living there, whose organism is producing proteins that are useful for you. So it's another layer of complication if we speak about the human proteome. So basically, it is now 20 years ago, the first human genome was sequenced. That took 15 years, lots of technological developments that made the project much shorter, but roughly it cost at that time $3 billion US dollars. And when the people launched the Human Genome Project back in the early 85, 86, they would say, oh, okay, because they were looking for funding. You'll see that will be tremendous. We will identify all the human gene and associate the gene to pathologies. And basically, if you look at the human genome, now it's still on this shape. It's a draft, elaborated draft. We are something like 30, more than 35 version now. And it's increasing, the complexity is increasing, it's revealing. But basically, how many genes did these people associate to pathology? Roughly 100, no more. Why? Because in fact, as I said, genes is a linear sequence of 4 letters, you know, and it has no other function than encoding for protein. And who's holding the biological function? These are the proteins. So the proteins in a patient's disease cells, all the key to understanding and maybe to personalize medical treatment. So we have to look at the protein. And this is a slide that I borrowed to Neil Kelleher from Northwestern University in the US. Only protein can tell. Indeed, take an example, the tau protein that is known to be involved in Alzheimer's disease. The gene doesn't do anything. The mRNA doesn't do anything. And it's the hyperphosphorylation of the tau proteins that is the trigger to see the disease expressing, you know. And we have plenty of CRASP4B, for example, in cancer. There's some hypervariable region of the proteins that are triggered, some tumor-specific proteoforms, but the gene has nothing to do with it. The difficulty when you look at the proteome, so this is another important slide I present to my students. If you look at genomics, this is what we call the world of possible. You look at the transcriptome, it's what's going to happen, and maybe it's not sure it's going to happen. If you look at the protein, which is the world of proteomics, you're looking at what's happening in the organ, in the cell at this moment. If you look at metabolomics, what has happened? Remember, a metabolite A will give a metabolite B, because there's an enzyme working in between. And what is an enzyme is a protein. So the more you go down on this slide, the more the complexity is important and the dynamic range is used. And you know that in proteomics, we don't have RT-PCR. We cannot amplify rare protein. So we have to deal with a dynamic range, complex sample, try to get rid of abundant protein to see the teeny and discrete protein most of the people are interested in. So it's a big challenge here. So
hey, Charles, can I have a comment here?
Sure.
On the previous slides, I think the complexity of the metabolome probably is less than the proteome.
Probably in complexity, well, yeah, that's popular. Yeah, you're partly right. It depends on how you look at it. But basically, proteomics and metabolomics, you're at the same level, you know, for sure. But it's discussable, right? It's more complex than transcriptome and genome, indeed. But I would rather say that metabolomics it's a bit complicated, although it's easier to study compared to proteomics.
Yeah, it's easier. Okay. Got it.
So back in 2010, the Human Proteome project was launched. This is the website page on the HUPO website. So the Human Proteome project is an international coordinated effort designed to map the entire human proteome in a systematic effort. Okay, so it's divided into two main initiative, what we call the CHPP. So it's a chromosome-centric human proteome project. So every important country has adopted one chromosome, not to work specifically on that chromosome, but to take care of the annotation of the protein from that chromosome. So basically you exchange information between PIs of every chromosome initiative. So the idea is to characterize every single protein corresponding to every single gene on one specific chromosome. Or the other part is a BD, biology disease HPP, where you focus on important biology research aspect and you look at some proteins that are known in the disease you study. For example, you work in the liver, you know a few proteins that are important for the liver physiology and the pathology, and you study these in the context of the pathology, so which is quite important here. So there's actually a large bunch of initiatives. There's a revival at the moment of initiative because some disappeared with the leaders and some new arrived. So there's a new initiative on the website that just started, and there's a cancer one from China at the moment. I just updated the website last week. Basically, there's an HPP matrix because all this work is supported by pillars. So MS is a mass spectrometry pillar. You have pathology, which is the last one, the pathology pillar. KB is the knowledge bases. ABS is antibodies. And basically, if you work on the HPP world, you will rely on all these tools here. This is an example of major integrated resources. The most important one, I would say, is proteome exchange. If you produce a proteomics data set with your manuscript, you submit your manuscript to a journal, it's required. So not every journal, unfortunately, at the moment is convinced that it's important, but basically every single journals in the proteomic world, it's mandatory to submit your raw data sets onto proteome exchange. Why is it mandatory? Because you can give access to the community to this work. If you publish a work with proteomics and your data set is hidden someplace or kept in a drawer in your lab, it makes no sense, because you can reuse the data set produced by others, ask a different question, and re-exploit this data set to produce new information. So it's very valuable. So it's connected to Peptide Atlas, which is in Seattle, and Peptide Atlas holds what we call the transproteomic pipeline. They will look at your data sets and confirm whether the protein you identify were really identified. So you made no mistakes, etc. There's also Massive in San Diego. It works the same like Peptide Atlas. These two work together. And once a year, roughly end of February, early March, they come up with the actual release of the data sets. and they contact NextProt in Geneva in Switzerland, who's updating the number of proteins from the HPP. And you have, for example, Human Protein Atlas is a different approach. You look for an antibody against a protein, human protein, go to Human Protein Atlas. Don't spend time looking for crappy antibodies for whatever companies. You go to the Human Protein Atlas. These people have in mind to produce specific antibody against every single human protein. So you have billions of immunohistochemistry. You have plenty of information because it's linked to NextProt, to Peptide Atlas, to Proteome Exchange, to all the transcriptomic RNA-seq datasets. So it's quite important. There's plenty of other tools, but these are the major ones that you can use when you're working in the HPP field. Back in February 2020, we succeeded in taking the opportunity of the COVID period. We all knew, and we all know we could not go to the lab. The only thing we could do is we went pretty good at Zoom and Skype and all kind of team stuff. And we were all able to work remotely. And we took this opportunity to say, okay, let's write the first blueprint of the human proteome. So that was published in Nature, February 2020. And at that time, we completed 92.8% of the human proteome based on the number of coding genes we would know. Everything was reported in NextProt, which is the major database for the HPP. So the NextProt is headed by Lydie Lam in Geneva. And this is the new release because it was the updates of the HPP data was done at the end of April. So actually, there's 1,381 missing protein. Just to remember the concept here, every single protein is related to one evidence degree. PE1 is a protein that you have seen, by mass spectrometry, which is the absolute standard. PE2 is a protein that you've not seen, but you have seen the mRNA. PE3, it's inferred by homology. You've seen, for example, the protein in the mouse, and it's probably a mechanism which is well conserved, and you expect to see the protein in the human, but you have not seen it for the moment. PE4 is not, it's a potential coding genes, but you've never seen the mRNA and you have not seen the protein. And PE5 is mostly errors of annotation, it's uncertain. And basically, what we call the missing protein is PE2, 3, and 4. These proteins are the target. We have to look at them. And why did we did not see those missing proteins? It's probably about their very low abundance. They're probably time or stress dependent. They are tissue or cell specific. So if you don't look at the proper tissue, you will never see this. They may have some particular physicochemical properties. They might be unstable. And most of the time, all of these consideration here are merged. You may have a low abundance protein with a very unstable and with a teeny frame of expression within a couple of hours. And if you don't take the proper sample at the right minute, you will never see this protein. So basically, this is the new number. So we're now at 93.01% of the coverage of the human genome in terms of human proteins. So we're still missing a few. And basically, we have some guidelines. There's a committee that belongs to the HPP world. And every 2 to 3 years, we come up with new guidelines for the identification of missing protein. And what we could see here is that we have a plateau. These are all the previous guideline version. The last one was 3.0 that was written in 2019 in France, in Saint-Malo. We gather all together to work on that thing. And you see a plateau here. The number of missing protein is going to a plateau and is becoming very, very difficult to see this protein. And not scientifically because of the guideline, but the guidelines are so strict that it doesn't help a lot, And the idea is not to release the guideline, to make sure that all the identification are pretty good. But basically, we're coming to a world where the missing protein now are most of the time small proteins. You may have difficulty to find the requisite number of peptide for the identification. They might be expressed in rare tissues, etc., etc. So I'll give you an example of what I'm doing in the lab. I'm working on spermatogenesis. So these are all the people I will acknowledge that participate to the work I will show, the founders and everything. And I'll explain. I'm working on the testis. I'm a specialized for 35 years in spermatogenesis. So it's a complex phenomenon that includes the expression on a huge number of testis-specific genes and proteins. And I'm looking at the sperm, human sperm. You have to remember that when the sperm leaves the testis, it's not motile and it's unable to recognize the oocyte. Unable. And it will acquire this capacity during the transit to another organ, which is called the epididymis. So in the superior mammals, it takes something like 2 weeks, 10 days to 15 days roughly. And during this transit, there's a lot of sequential modification on the sperm, on the surface of the spermatozoa, lots of proteins secreted, reabsorbed, modified, etc. So it's quite important. And basically, years ago, the group of Lydie Lan in Nextprod, they say, well, they were looking at that time at the remaining something like 3,000 missing protein or more. And they say, well, that probably we should find these in unusual organ, for example, the testis. And then the year after, Matthias Hulen and collaborators from Human Protein Atlas say, well, that's great. The largest number of tissue-enriched gene is found in the testis. I was smiling. When I saw this work, I said, well, we go back to 2005, so 10 years before, we already knew that the number of specific gene in the testis was something tremendous, and there were probably a lot of specific proteins. And we said, well, most of these proteins belong to the germ cell after meiosis. So it's difficult, as you can imagine, to obtain human testis. There's no volunteer. If you are a volunteer, you just give me a ring or send me an email. No, I'm kidding. So the most efficient way is to work with the final product of the germ cell, which is the ejaculated sperm. So we teamed up, and the context is we're not doing a catalog. When you're working on missing protein, you contribute to cataloging the human protein, but you not do the work to do the catalog. You see what I mean? So there's a biological context, a pathological context, and all those teeny discrete proteins, if you have not seen them before, they're probably very important for the physiology of the organ and then the pathology. So the context of our work is male infertility, which is a high demand at an international level, and the sperm decline that has been published in different countries, and increasing number of testicular cancer that has nothing to do with the age. It's more linked probably to the environment, to plenty of things here. So it's quite important. So basically what we did, we teamed up with the chromosome 2 team, which is the Swiss people from Nextprod, which is even better. We speak the same language. And we could first design a method to roughly analyze missing protein properly. And I'm quite happy because that work here by Christine Carapito and collaborators, that was back in 2015, is now used as a standard for the identification of missing protein. So we established the same year first proof of concept that the sperm was a really good cell to identify missing protein. We identified the first bunch of 83 missing proteins. The year after, still published in the JPR special issue of the HPP, we identified an additional number of 243 by a deeper analysis of focus on the membrane of the sperm. The year after, again, Christine Carapito, first author, we identified another 12 additional missing protein by using a PRM approach, a targeted approach to look at protein that we knew it was there, but difficult to catch. And the story went on and on. Every year we publish a paper. There's another one at the moment in preparation in this JPR special issue. So basically, that's an example of what we do for missing protein. But your data is not only focused on identifying the proteins. It might be useful for explaining the biology of the object you're studying and the pathology. So the idea is to look at your data with a different point of view and think integration. Basically, if you want to do integration, you have to rely on plenty of tools. And the difficulty is that there's no universal approach. I mean, the limit here to use other tools is the imagination of people. Okay, if you're pretty smart, you may have the idea to use that and that tool, and then you have to deal with experts and find the people that will help you to do it. For example, we teamed up with the Human Protein Atlas, the people, so Cecilia Lynchcock and Feria Hikmet, and they say, okay, you guys are working on the testes. We have billions of immunohistochemistry image. Can we export and extract some information, functional information, by just looking at the image? So we say first, okay, let's take 500 protein really expressed in the testes that we all know with distinct patterns and try to look at the anatomy here and the expression of this protein. So they designed for that. That's an extract of my personal screen viewer, of HPA. I have all sort of information here. If you're an ordinary visitor, you will not see these. So I'm using it to annotate properly all the image, provide some information on the dilution of the antibody, etc. So I've done it myself for months. It takes time. But basically, looking at the expression of protein, you can come up with some information, for example, proteins that are shared with other organs. So the more it's blue, the more it's shared with other organs. Proteins that are specific to one germ cell at a specific stage. Or you can come up with proteins that are with shared expression from a certain stage of spermatogenesis. And basically, if you look at all the image here, we come up with some clues on possible function. And among the missing protein we've identified that were in the bunch of 500 here, there's some proteins that are possibly involved in meiotic checkpoint, prevention of abnormal meiotic recombination. So everything that goes with the germ cell maturation. But then if you look at other image from HPA, not only the testis, we looked at, now you focus on one protein, for example, and I give you two examples here. One first protein is called CFAP45. We don't know the function, okay? We've identified this missing protein in the testes. And if you look at immunohistochemistry with the antibody they have developed, if you look at this protein here, was expressed in the flagella of the germ cell. Perfect, that makes sense. But if you look at the fallopian tubes, you have some teeny staining here, and the staining in on ciliated cells from the fallopian tubes. So you look at another one that's a known protein for years, FCAP-B1. It was expressed in late germ cell with the flagella and what we call the late spermatids here. And you have another staining in the ciliated cells from the fallopian tubes. So basically the idea, cilia, flagella, there's not that many difference. They're probably the same mechanism. So probably this protein with unknown function here, we just identify as maybe a role in the flagella and cilia formation or functioning. And basically, a year later, we saw this pretty nice paper speaking about the deficiency of CFAP45 in both the human and the mouse with a knockout. Basically, if you're a human, you have a CFAP4 deficiency, you have a situs inversus totally. That means all your organ are the opposite. You know, it's a rare disease. It's not a disease. I mean, your life is not in jeopardy, but you're all opposite. Your heart is on the other side, and etc. etc. But if you do the knockout in the mouse, you have an astenozoospermia, which is the control mouse sperm with the beating of the flagella. You have 70% or they don't move anymore, and 30% are totally still. So the sperm doesn't move. So probably, and we're sure now, CFAP4 is involved in the flagella functioning. And this is the sort of information we wait in the grand project I will talk about. Now, we need to connect a protein with a possible, at least one function, at least one. And you know, protein in the human have probably a tremendous amount of function, depending on where they are in the cells or in the cell next to it. That makes really a difference. Basically, to go further, because I've done this by hand, we teamed up with a group of people in the UK that are in artificial intelligence, and say, well, everything that I did manually, could you develop some sort of algorithm that you could annotate the image automatically? And it took a couple of years to discuss with these people and to understand each other. So this is always interesting to discuss with people that are not in your field. You have basically no chance to discuss with them. But if you can manage to understand each other, as we say in France, is the mayonnaise working? That's a rupture thing. You can come up with very nice development. And they develop for us a tool that is annotating automatically the image with a percentage of diagnostic performance over 96%, which is fantastic. So basically, we continue with this missing protein, but just an interesting thing that is going on in my lab at the moment. Among the number of missing protein we have identified, 7, the genes is not expressed in the germ cell. So how come? And basically, if you look at RNA-seq tissue expression in the testis, epididymis, prostate, seminal vesicle, the human reproductive male organs, but you see that these 7 proteins, the gene is expressed in the epididymis. What does this mean? That means that during the transit through the epididymis, the protein goes and is secreted and stick to the sperm. So this is, for example, one is a missing protein, defensin B132, is expressed in the mRNA expression in the epididymis and in a few other little things, a little bit in the prostate or adipose tissue. If you look at there's no staining beside background in the testis, and this is the head of the epididymis. So the testis is connected here. And you see the transiting sperm is already stained, heavily stained, and the pattern of secretion of the protein by the epididymis. And basically, this is the expression of the protein with a little viewer here. And it's mostly expressed in the head of the epididymis. So this is what we try to understand now, was the use of this protein for the maturation of the sperm. And there's another project that I just started with, published a paper last year in the special issue of JPR. One of the students is following the human embryo, the development of the human embryo from the zygote to the trophoblast and the implantation stage. And she's looking at different stages of the cells and trying to find very discrete missing proteins that we know are expressed in this sort of cells. So they're doing iPS cells, and they've developed a model of blastoid, which is some sort of in vitro blastocyst. And these are, for example, 20 missing proteins that can be expected in naive stem cells or in trophoblastic stem cells. And basically, we get the cells in culture, extract the peptide, do the proteomics, And we were able to identify a few missing proteins. So the difficulty here is that we have the guideline, and for identifying missing protein, you need 2 peptides of 9 amino acids, non-redundant, non-overlapping, and this is quite difficult. So sometimes we have 1 peptide, we have only 1, and the number of copies of the protein is low enough that you're not able to identify this protein. So we have what we call one in wonder. We have only see 1 peptide, not the other. So this is the difficulty, and we still continue. For example, we look at one protein at the moment. It's called TRIM61, which is expressed in the morilla of the developing blastocyst. So it's quite of interesting. So as I said, we don't look for missing protein to do a catalog. We contribute to the catalog, but the idea of the work is to understand all cells here are directed towards one fate or another, identify key proteins that are involved in, at this stage here, the human preimplantation development, and use this protein as biomarkers to be used in a non-invasive method for embryo selection. And the whole work is for better IVF results here. So basically, 2 years ago, we announced the Human Grant Project. So we build upon everything that was done, the resources that were constructed, the effort we've done in the HPP, and say, okay, we have a bunch of remaining protein. It will probably take 10 years to identify because they're pretty discrete, difficult to catch. But we can go to the next step, which is now to correlate every single protein to at least one function. And the idea is to understand the proteome in the context of networks. The idea is not to construct something from scratch, is to partner with complementary consortia and initiative from different people, but try to interest these people in the HPP. Plenty of biologists in the world don't even know what is HUPO. I've barely heard about the Human Proteome project, but they don't know a word about it. Plenty of biologists have never used mass spectrometry, for example, so it's another world for them. So the idea is to attract these people by saying, okay, if you guys provide information on the function of your favorite protein, your favorite family of protein, your favorite pathology, come with us and share your data. If you share the data, you will get other data from other groups, and it could be useful for your own research. It's a forward-thinking strategy. We try to construct the project on the fly because we have still no idea how difficult it would be, how to attract people. And the idea is to be a focal point for translational researcher, for mechanism, diagnostics, therapeutic. The main thing is to improve indeed human health. So Rob Morris, who was chairing the HPP before me, wrote a white paper that you can access from the HUPO website, and I recommend you access this paper and read it. So the grant project is open to all interested group or individuals. We don't want to impose a way of working. We want people to use the resource they are familiar with, the strategy they have always been using, but try to find a way to share the information with us. It's agnostic to disease and biological context. Everything is important in human health. So whatever the organ you work on the pathology, your specialist of, the idea is to have the knowledge of system being worked on through the access to resource to perturb with systems. So build networks and understand the context of each protein to build a knowledge base of overlapping network. And probably NextProt will play a key role here. This is an example of people from Toronto you probably heard about Target 2035. They use chemical probes to perturbate a cell line, and they do some comparative mapping with or without the molecules. And you look at the interactome, but not on the way you classically look at the interactome. You try to exploit some possibilities and define what is interacting with the interactome, what is a perturber of the interactome, to understand the impact of the molecule and the function of this protein. So I've done the same in France with the equivalent, which is ChemBio France, which is a national chemical biology library. So these people are developing small molecule for human health. You know, basically all the big pharmas have access to this catalog here. And we launched a teeny call that was I have a Zoom tomorrow with one of the PI here. So you come up with your favorite cell line. You use the molecules at different doses that we decide it would be 0.1 EC50, 1 EC50, 10 EC50. So you're pretty sure you have an effect here. We do the proteomics analysis, look at the interactome. So the idea is you come up with some data that will help the team to understand the biology of the cells and the molecule they're looking at, because they're interested by the molecule. ChemBio France, you get some data here that will enrich your catalog and the information you can provide to users about that specific molecule. So these people can publish their paper. And then there's an agreement here that the Grand Challenge can benefit for all the dataset produced to exploit further the data. So at the moment, it's a teeny pilot project. There's 4 projects on 4 molecules. There will be another call in September with another 4 molecules or 4 groups. And we're working on MS method and data acquisition. So again, we're on the fly trying to understand how we could make something really good with the proper control. How shall we exploit the data? We need to set up a central repository for all the data sets. So everything is open. We don't have any clear idea on how we're going to do it, and we'll do it on the fly. So basically, it will be probably my last slide.
Hey, Charles, another question for the previous slides.
Sure.
ChemBio France. So in this project, which cell line did you use? Or do you
so basically, I don't remember the cell line. It's very different cell lines. I can look at it. I don't have it in mind. I should take my lab book here, someplace here, and look at the project. So I cannot.
You are using multiple cell lines or just one cell line?
No, there's one cell line. So basically, you have one cell line which is responsive to the molecule, and another one. Ideally, if you come up with another cell line which is non-responsive, that makes a good control. But I can send you the information later and give you the example of the cell line that are used. They're not dedicated to cancer, to whatever. It's really specific. I remember one molecule is in the field of neurology, so it's a cell line that is pretty responsive. So, but I don't remember. I don't want to say mistakes, and I don't remember the cell. It's very, very different. Very.
But then, did you try different time points?
Yeah, well, the people have already an idea of the time where they have an effect. Okay, so they already agreed upon one specific time, but we use 3 doses to make the whole thing. So basically, I know it's between 10. One project is something like 10 hours. The other one is 20 hours. So these people already have some knowledge on the effect of the molecule and how long does it take to give an effect. So it's accessible on the ChemBio France library for every single molecule. So you have all the information. They already know about the effect of the molecule, what is the main target and everything. So it's important. So basically, to submit the idea is the main problem with the Grand Challenge. It was the problem already with the HPP. There's no specific funding in most countries. All these people are working on their own money from the lab, including myself. I mean, I'm using money from another project to manage to do a few things. So basically, what's missing is dedicated funding by countries. So again, I'm French. So what I use French as a testing ground. So I contacted the French ANR, which is the main funding body in France where you can apply for funding. So every October, you have a generic call. Whatever you're doing, I mean, you're a physicist, a chemist, electronic, mathematics, or biology, you have a call. You explain what you want to do, and you expect to get some funding. So basically, I discuss with these people, discuss on the biology side of the call, and I was accepted to help writing the call, the text of the call. So in France, people on the last call, it's under evaluation at the moment. They would see HPP in the keywords and everything. So I don't have a report now of the number of projects that are funded. We will know it early in July to know whether these people were financed. So basically the idea, I mean, we created a small template on the HPP website. There's a group of people, a task force, which are mainly all the people in the HPP executive committee. We look at the letter of intent, a few words, the keywords you're providing. We're not here to say your project is good or not good. We're here to provide you with recommendation. And then you go submit your grant application. And the idea is to provide you with a letter by UPO and HPP saying, okay, the work these people are presenting to their national funding agency fits perfectly with the objective of the Grand Challenge. We support it. That doesn't mean it will work, but at least we will send a list of projects that are some sort of accepted by UPO because they're relevant to the Grand Challenge. So then it's the national funding agency who will decide whether or not they will fund, which is another problem. So we'll see this year in July whether it worked or not. And the idea is to do it in different countries. At least in Europe, we know we all work the same for every national agency to fund the whole thing. And then last year in Cancun, we were presented that wonderful Pineau project by Tianan, Fuchu, and all colleagues. And to me, I don't see how we can work together, but we have to work together, which is incredibly important. And you people in China have a fantastic view, a great funding, and grand task force to work on it. And I'm really hoping that we can come with something very, very serious to help human health and pathologies. And if I have to convince you with another image, it would be my last. This is the one I like with my colleague Tianan in Cancun last year. And we will probably work hard, try to work together and build something on the Grand Challenge and the Pineau project. And thank you for your attention. And if you have questions, I'll be there to ask and answer your question. Thank you.
Thanks, Charles. This reminds me of a very good time last December. So can I ask one burning question? So
sure.
You mentioned we welcome others to contribute to the Grand Challenge project. Ideally, the biologists from different fields, and they have their own research interest, but if they can collectively contribute to the Grand Challenge project, they can also get a benefit from the database. So can you maybe elaborate a little bit more? So as a biologist, let's work on sperm or work on liver or whatever process. What exactly should they contribute? For example, if they contribute chemicals or they contribute some samples with perturbation or whatever knowledge or sample or whatever thing or data or whatever data, should they contribute the Western blot or what can they contribute?
Well, the different layers here. Well, as I said, the main difficulty is the funding. So basically, the first way is to get specific funding to develop a project whose aim is to really address the function of a protein. That's clear. On the other side, some people have already some data that they do not share, probably because they don't understand that it could be valuable for others. And I'm thinking about clinicians. So basically, we're working on human health. We're doing some very fundamental work, identifying proteins, seeing vitro system, how it works and everything. But probably on the other side of the campus, you have some clinicians working on the specific pathology, and that specific protein you're working on it makes sense to them, you know. So the idea is to attract the people. So we still don't know how to do it, but the idea would be like in a first layer, there's plenty of possibility. But the first one would be some sort of Wikipedia information. Okay, you provide information on everything you know about this protein. And then as a reward, you will probably then have access to other data provided by other people. So the idea is to, and I'm thinking on the fly again, is to start from scratch. We need first to set up some sort of primary repository, see how it works, make the tools to make these people provide information and etc. And then if they have integrated research project where they really come with information on the network of protein, and then they could be, it could be easy also for them to upload the central repository with that information. That means then that you need to have some curators, you need to have some people to take care of the database, you have to have some people that would extract also knowledge, you know, that's basically what you aim to do with the PIA project. So everything has to be constructed, and the first one to start will be the examples, and then we can think about other ways to provide information and etc. So it's really forward thinking on what we should do with the project.
Okay, so now there's a lot of possibilities for people who are interested, biologists, clinicians, or computer scientists. If they're interested, they're welcome to contact Charles.
And as I said, it's almost a joke, but some biologists don't even know anything about the Human Proteome project. They've basically read it a few lines in a paper. They've never been interested in that thing because it's not the primary subject. They don't care about it. And some people don't even know anything about mass spectrometry, and they would say, well, protein is too difficult. I'm working with the mRNA, it's easier. And that's basically, that's sad, but most of the people I know are thinking that way. Biochemistry, too difficult. I had bad time at the university. I don't want to hear about protein anymore. But they should have, they should consider that protein is the future. You have to work with the proteins. mRNA is not enough, you know?
Sure. So, Changcheng, you have any comments, questions?
Yeah. Hi, Charles. Great talk. And it's very, very pleasant and promising project to me. And here I have two questions. Did the grant project, charity project, provide SOP or protocols to the groups who are applying to join, and did we have to apply the same SOP, or we can have different SOPs for data processing and data analysis?
Okay, so at the moment we're just thinking about it. We have the French proteomics meeting in a month from now, and the whole group with Cambio France will be here, and some of the colleagues from abroad. And we have to, the idea is to think about standard operating procedure, the proper control, how we will do it. For example, with mass spec, are we going to do DDA, DIA? we have to make sure that we're all using the same approach. If not the same instrument, would be great, but everything will be constructed on the fly. And we hope that for UPO 2023 in Busan, I'll be able to come up with a first data sets and show the people what we could do with it. The most important at the moment to me is how should we analyze the network, the interactome that will come up with proteins with level of expression. So we should not look at basically, okay, this protein went up, this protein went down, and the technique to approach the protein that are interesting is more the wet finger. Say, well, I can feel it well here, this one is interesting. No, we have to make some sort of, and it's purely computer machine learning and stuff like that, how we can come up with some prediction. Okay, that molecule will affect that pathway, for example, meaning every molecule from the same family will probably affect the same pathway. What are the proteins most important that are affected by the molecules, and come up with an idea on how they would function, what they serve for in the cell, but we have to think globally. And I'm not a specialist of this, but I anticipate, but we have to look at the interactome differently than what we do at the moment. The question is...
I have a comment here. So personally, we are more using DIA. I think DIA is very promising, but it will be very difficult to ask everyone to use the same type of data acquisition. Even for DIA, there are different instruments from different manufacturers. And over time, let's say 5 years later or 10 years later, and the mass spec will be different. But also the data may be different, but the interactome should be consistent.
You're right.
See, under some perturbation, under some chemical or genetic perturbation, some network increased, some protein up, some protein down, and they are interact with each other. And this network, they are consistent. So we should have some uniform data format for this.
And I have involved Sue Wentrobe for the MS pillar to think about it, and we came up with we had a discussion last week about it. Say, well, DDA, DIA. Well, a group can perform pretty well DDA because they master the technique perfectly, rather than launching a DIA approach that is not already optimized in the lab, and you come up with crappy results. So, well, and basically the difference between DDA and DIA, you'll come up indeed with additional identification of protein. But basically, if you perturb a cell with some molecules, the proteins that will be the key players here are probably highly expressed proteins that are accessible easily without the need of DIA. So it's an open question at the moment. But you're right, the interactome will always be the same. So basically, with the technology evolving, okay, you do the same experiment in 2 years, you will come up with additional identification. But well, so what? Does it make sense? I'm not sure about it.
Yeah, can't agree more. And by the way, I think analyzing interactome, the methods is need much. We need much advanced methods such as systems biology methodology.
Yes, absolutely.
System view, yeah. And since AlphaFold is very success in recent years, and there is a paradigm from protein sequence to protein structure, then to protein function, right? So basically, this paradigm was proved in structural biology, but in proteome science, I think there is also a paradigm for from protein mass spec data to protein abundances and to protein functions, and we have much work to do about this. Yeah.
Yeah, and basically it's an open invitation for you guys. I mean, we come up with this teeny pilot project. We'll come up with interactome datasets. I mean, the idea would be free to explore the data sets and see how you can develop an approach to extract information. You know, it's open to everyone. This is the idea. My idea is to construct the consortium on the fly and say, okay, some people are interested because they have a specific knowledge to exploit the data set. Okay, I want to be part of it and try something. And probably we'll come up with some standardized procedure, something really serious that has pretty much chance to provide the relevant information we're looking for, you know?
Yeah, and I also have a basic question, maybe asked by the top-down proteomics researchers. Since Neil proposed the proteoform atlas, right, 2 years ago, and he think for proteome we should discover more proteoforms as much as possible. And for the grand challenge project, we focus on the protein functions. How do you combine these both of them together, or how do you consider the philosophy behind this grand project and Human Proteome Atlas?
Well, it should be involved in it should be part of the grand project. So I've not discussed with Niels and his colleagues specifically on that approach, but to me it would be a strategic error to launch something totally separate. You know, we all have to work together with a common goal. And the world of proteoform is huge. I mean, it's kind of difficult, but as Tianan said, the technology will evolve, and I'm pretty sure in 5 years it will be rather easy to address proteoform information. And they go pretty fast. And I know some constructor like Bruker, they're full speed on that, you know, because it's an expected way of analyzing protein, not to do bottom up anymore, you know.
Yeah, so I would like to see that it can be considered as two sides of a coin. Is that right? Yeah, thank you.
Sure.
I approve, Pineau. I have 4 questions. First, thanks for your excellent introduction about missing protein and HPP project. Yeah, I'm very interested in the testis specific isoform you mentioned. But how to define the specific testis specific isoform? Because you in my I didn't see you have compared with other organs.
Yeah, well, so this is known for years. You have two groups of, I would say, proteins, speaking about proteins, not the genes. You have proteins that are expressed in plenty of organs, but that are highly expressed in the testes. So you can find them in the brain, mostly in the brain. You have this brain-testis correspondence. It's always a joke in the world, but it look alike, you know. And you have plenty of proteins you can find in other organs, but in the testes, for some reason, it's highly expressed. And then you have another group which can only be found in the testis and not in every single cell. It's found in the germ cell. And if you think about it, and why, is by these germ cells do not exist in any other organs. You cannot find a spermatid or a sperm in another organ. So all these proteins that are really specific to the germ cell line, especially after the meiosis, where the cells is N, is not 2N, is haploid. And it gets rid of plenty of material to develop a flagella and everything. So this is very peculiar. And all the proteins expressed here are really highly tissue specific. So you cannot find them anywhere else. And then also, if you look closely on some proteins, you have some proteins that are expressed in another organ. And you have an isoform in the testis with a few amino acids that are missing. You don't know why. The protein looks the same, does the same role, but for some reason it's slightly different. There's no explanation except that probably, so it's one of the possibilities, not sure. The testis works below a certain 5 degrees difference with the rest of the body. The testis works at 32 degrees, not 37. So maybe by some protein with a few missing amino acids, it works better as an enzyme activity, for example, at a lower degree. But wow, that's questionable. You know, that's an hypothesis that's been going on for years. We don't know why.
Okay, I see. So this.
Sorry to interrupt. I have a follow-up question. Did you consider de novo sequencing for these specific tissues, such as testes, for missing value, for missing protein discovery?
Yeah. We launch a few things like that. So far, is not successful, but and we use also a proteome genomics approach. And we were able to characterize a couple more proteins that were missing, you know, that were not considered relevant to coding genes, you know, and you find a peptide and say, well, finally, this is a coding genes. And for example, we came up with some proteins that were classified PE5, so considered to be an error of annotation of the genome. And from PE5, they went up to PE1 because we saw the peptide and it was a new protein. For which we still don't know what is the function, you know. But it's not hundreds, and it's a couple of proteins. You count them on two hands, you know, no more. For physiological mechanism like spermatogenesis, it's conserved between all the mammals. The idea that you can come up with new proteins that were never discovered, I don't believe it. So there's only a few proteins that you can identify, not thousands. You know, like some people pretend they would do.
Okay, thank you.
Sure.
Please go on, Siri.
Yeah, this is my second question. Is about the target method to identify these missing proteins.
Yeah.
You mentioned this target method is to identify some missing isoforms.
Yeah, it was to identify. No, the idea was to say, okay, if we cannot identify some missing protein by just looking at the sample, okay, we have some, we go deeper in the sample. For example, we prepare membrane, we prepare things, and then we amplify the discrete protein. Okay, and if it's not enough and there's still some protein that you cannot access, the idea was, okay, why don't we use a PRM approach or MRM approach and say, okay, we're going to look at that specific protein with a targeted mass spec strategy. Okay, so we come up, we purchase some peptides and everything, and we spike the thing, and we're trying to do MRM and PRM to identify this protein. So we were targeting something like 38, and we finally identified only 12. Because we would see only one peptide or see no peptide at all, meaning that those proteins were teeny, very, very low expression level, not accessible in the condition of the analysis by the mass spec. So we continue trying to find additional one, but well, it costs a bit of money also. A PRM approach that's not free.
But I saw you mentioned at least 4 peptides have been identified by the targeted method. You think this is you have identified this missing protein for at least how many peptides?
Yeah, you're discussing about this the work on the embryo, the stem cells. Yeah, we have we have identified. Yeah, on this embryo stuff, we have identified only 4 missing proteins, and but we have 6 other one-hit wonder. Remember, to identify a missing protein, you need 2 peptides of 9 amino acids each. So if you have one peptide of 7 amino acids, forget about it. It's not a fit with the guidelines. So you suspect the protein is here because 7 amino acid is a pretty decent peptide. but you don't come with the guidelines, so forget about it. So you have to make some effort to concentrate your sample and eventually use targeted approach to catch this protein, because you know it's here, but you don't have a proper validation.
I see. Yeah. Do you have made the situation when you identify the missing isoform? You have now for this protein, for this isoform, it did not have unique peptide to identify.
Oh, yes, actually, we have this case. Basically,
so how to identify this and this? Yeah.
That's a main problem, and at UPO in Busan in September, I will call for another guideline meeting. You know, we have to make the guideline evolve. For example, plenty of the missing protein, I told you that 1381 exactly at the moment. There's more than 400 is our olfactory receptor. Nobody has seen an olfactory receptor in the human. We still always see in our sample testes, we always see the same peptide, one peptide, but we don't see another one. So not enough to identify this olfactory receptor. These are rare protein. And then there's another bunch in the 13 something. These are small peptides, small molecules that belong to the defensin family, for example. Defensin is all these teeny proteins, though the average is 40 amino acids. So if you need 2 peptides of 9 amino acids, forget about it. And basically, so what we doing at the moment? To do this digestion of proteins, you use trypsin. So you can use another enzyme to other cleavages to get other peptides. The problem is for this defensin, there's no cleavage site for any other enzyme. So basically, you can use trypsin. You will have, for example, I think about one defensin, you have a peptide of the defensin, it's 42 amino acid. You can get one peptide of 21, and that's it. It's not enough, but we're sure the peptide, the defensin is here. We're sure, and we have the data from Peptide Atlas. These people have developed an antibody, and you see the staining, you see it's here. So we have to make the data evolve, the guidelines. Otherwise, we will not be able to officially identify those remaining missing proteins.
Okay, I see.
But not release the guideline too much, because otherwise we may come up with crappy information, you know. So that's a difficult thing. And then there's other ways of analyzing the, because basically the guidelines are based on DDA approach. So if you come up with a DIA, well, you don't fit with the guidelines, but plenty of people are using DIA now successfully. So shall we make a new version of the guideline? To me, it's important to discuss this issue with the colleagues and make sure we come up with a proper thing. Otherwise, those missing proteins will remain missing for decades, you know?
Yeah, I see. So the next question is about the image. You use the image to predict the protein function, such as IHC. But if you meet a membrane protein, how to confirm whether it's a channel protein or a receptor protein?
Well, that speaks perfectly with the objective of the grand challenge. You have to be imaginative enough to develop some sort of an assay to demonstrate that it's a channel protein or it's a receptor or something. So you have to imagine the whole thing and come up with an information when you have one. Now it's wide open now, you know.
But in the IHC, did you have see the you can see this function?
Yeah, not yet. I mean, we the student is working on it, so we'll probably come up with some nice information by the end of the year. I submitted a paper here.
Okay, I see. I will read this paper. And the last question is about the Camel Bell France this project. Yeah. When you use EC50 to treat the cell line, but if the drug is non-responsive, how to define this EC50?
Yeah, you're right. So the idea is to use the cell is not responsive. You use the same amount of the amount of the molecule. Similarly, with the responsive and the non-responsive cell, this is the only way to do it. Otherwise, you're right, the cell is not responsive. So whatever the concentration of your molecule, you won't have any effect. So it's used to be it's not mandatory to come up with a non-responsive cell line. But if you happen to have it, we add it in the analysis, so it makes an internal control, you know.
Yeah, and
probably after analyzing the first project, we say, well, finally, the non-responsive cell line doesn't add anything to the story. It's more work than we need, and maybe we'll skip it. But at the moment, the ChemBioFriends people say, well, we would like to have a non-responsive cell line included in the analysis. We'll see. Again, it's a pilot project, so we'll see by the end of the year whether it's a successful approach and we can build on it and amplify the project with other molecules, other cell lines, etc.
Yes, you are right.
Groups working on specific, pathology or physiological approaches. So it's interesting.
Yeah, I have suggestions for this. But if you use the EC or IC50, you should do some experiments in the cell line first. But for some unknown moleculars, it's not easy.
Yeah, basically the people who answered the call and we selected the project, the people are already working on one specific thing. They're working, for example, on neurology. They already use some molecules or they know they had an effect, and then they go to this lab and say, well, this molecule in the catalog, I'd like to test this because it's probably better than the one I use at the moment in the clinics. So let's study how it works, whether this molecule is better. And that was an opportunity to launch this call and say, okay, if we can grab some information from these people, get their hand on their data sets, we may use it for the HPP Grand Challenge, and on the same time, these people will be free to use them for their own project. And ChemBioFriends will collect some additional information to strengthen the info in the catalog for other when they will come to get this molecule.
Yeah, so if you use the same concentration for each drug, it might be more easier to see more moleculars.
Sure.
Yeah. Okay. Thank you. That's all my questions. Yeah.
There's a question from the audience. Yeah. You can ask.
The question one is how to identify isoforms and how to identify the shared peptides of isoform of the same gene.
Oh, okay. Basically, for identifying a protein, not a missing protein, in general, that's pure mass spec and biochemistry. To identify a protein, you have to identify what we call a unique peptide. So it used to be called proteotypic peptide. That was more speaking to me. The term now is a unique peptide. What is a unique peptide? it's a peptide that is only expressed with one specific protein and not shared by another one. Okay, so if you're looking at an isoform, this unique peptide, and you may have several, can only be attributed to one of the isoform, not the others. So that's the rule. So if you have two very close proteins, because they change by a few little things, they are from the same family, you have form 1 and form 2. You have to find the peptides that correspond to either form 1 or form 2. If you cannot do it, and you're working on peptides that are shared by the two proteins, you will never know which protein you're working on. That's the problem. And there's a few cases where there's no way of distinguishing between an isoform and another one. We've published it 2 years ago in one of our papers, and we explained the case for one specific protein. We were unable to find a proper peptide, and we still did not know whether we were working on isoform 1 or isoform 2. I don't remember the name of the protein, but that was tricky. And we had Lydie Lan working specifically on it to make sure that we would not tell bullshit in our paper, you know, and give you an example of how difficult it could be.
Yes, thanks.
Sure. Or we can use a top-down proteomics. Thanks, Charles. Now the time flies, really. About a half hour. We learned a lot, although I know proteome very well, but you have done very nice introduction of the proteome. A lot of impressive numbers. I think probably we can also use in our future presentation to educate the people, biologists, clinicians who even do not know proteome and do not know mass specs. I think they should know this and they know the powerful.
Excuse me, there has the second questions from the audience.
I'm going to conclude. So the second question, how to determine if a missing protein is caused by identified method or from?
That's an excellent question. In fact, you cannot tell because if there's no, in mass spectrometry, there's no proof by absence.
Yeah.
If you don't see a protein, that doesn't mean it doesn't exist in your sample. It means that in the condition of your analysis, your mass spec were not able to identify the protein. It can be here, but you miss it. You cannot see it for plenty of reasons, technical reasons. Mostly is due to the dynamic range and the fact that your protein is very low number of copies. So it's hidden behind proteins that are really high level expression, you know. So basically there's no way to say whether it's the method or is the protein is really missing. No proof by absence is mass spec. That's the rule.
Yeah, I agree. So Charles today introduced the Grand Challenge project. This is for the next decades, 1 decade or 2 decades. The whole entire community probably need to address this question. This cannot be completed only by the proteomic community. We also need a cooperation from biologists, clinicians, data scientists, and many others. So this is still a white paper, although Maurice has written a white paper for this project, but it's still very open to more discussions. So I'm very happy that Charles introduced this in our webinar. So in the future, more discussion will going on in Busan and also in China. Welcome to you to also join our CA HUPO meeting after the Busan meeting. And we are going to have a lot of discussion about the Pi-Hub and how the Pi-Hub can cooperate with the Grand Challenge project. Last time Fuchu gave a presentation in HPP Day. This is a sister Pi-Hub and Grand Challenge project. So, but how to become a sister, how to cooperate, I think this is our future topic. Thank you, Charles. Thank you, Changcheng. Thank you, Sun Rui. So Sun Rui is also the major person in our group working on the Grand Challenge project. Hopefully, she will also meet you in Busan.
Hope so. Yeah, great.
Yeah. Okay. Thanks a lot.
Okay. Thank you.
Thank you. Bye.
Bye.
Bye bye.
Thank you,
Bye.
people. Bye bye.